and then we can jump right into it.
Thank you.
So Rebecca is a postdoctoral research associate working for Dr. Kate Baker at the Department of Functional and Comparative Genomics in the University of Liverpool. She is currently working on working computationally on large data sets of whole genome sequences of Shigella collected from Asia and Africa. And her main focus is applying various statistical methods to investigate molecular evolution, phylogenetics, and the epidemiology of Shigella. So yeah, go jump right into it.
Okay, thanks for that introduction, Adil. Yeah, so I hope everyone is doing well.
Oh.
So can you hear me?
Yes, we can hear you.
Oh, okay. Yeah, so I work for Dr. Kate Baker at the Liverpool University, and our group work primarily with Shigella. So we use a lot of whole genome sequencing for epidemiological analysis and also AMR surveillance and all the other fun stuff. So today I'm just going to be talking about some of the work that I've been doing in applying whole genome sequencing to help combat shigellosis in endemic settings. So a brief introduction, shigellosis is an infectious diarrheal disease that's caused by Shigella. So this is a genus of bacteria comprised of 4 species. So we have Shigella flexneri, Sonni, dysentery, and Boydii. And the route of transmission for Shigella is fecal to oral. So shigellosis is endemic in low to middle income nations, but we could also, it can also occur in high income nations as well. But these are usually associated with travel or sexual transmission. So shigellosis is currently treated and managed primarily through the use of antimicrobial therapy. But Shigella have a really high level of accessory gene plasticity. So this means they can really easily pick up resistant genes. And we know with the increase in antimicrobial usage, there's been a really high increase in resistance. And as a result, multi-drug resistant Shigella are really widespread across the globe. And currently there's no licensed vaccine for Shigella. So treatment and management for this disease is really limited. Whole genome sequencing analysis is really useful for epidemiological surveillance of Shigella, and it has been successfully applied in previous studies where the analysis have highlighted the importance of horizontal AMR transfer and in driving its epidemiological spread. But these studies have mostly been based in high-income nations. So they were either non-systematically sampled or they were sampled based on specific epidemic concerns. So they're not really representative of a population where the disease burden is the highest. The Global Enteric Multicenter Study, GEMS, was a large prospective case control study. It was conducted between 2007 to 2011. The study was funded by the Bill and Melinda Gates Foundation, and the goal of GEMS was to determine the cause of moderate to severe diarrheal disease in children under the age of 5. And the sites was conducted over 7 countries across sub-Saharan Africa and Asia. So these sites were selected because they broadly represent regions with the highest disease burden across the globe. And GEMS identified that Shigella as among the leading cause of moderate to severe diarrheal disease in children. So in the table shown here are the number of cases and control for all Shigella species in GEMS. And because GEMS isolate was systematically sampled, these are proportionally representative of disease in the population. So to help us to understand, to enhance our understanding of endemic shigellosis, we performed whole genome sequencing analysis of isolates from GEMS to look at primarily AMR prevalence, genetic diversity, and how the diversity may implicate vaccine development. We also wanted to look for genetic factors that were associated with disease because this is a case control study. And also, hopefully, this will help us to inform interventions and also management of shigellosis, where in a population is most vulnerable to this disease. So in this study, we only analyzed Sony and Flex isolates, as these two species were responsible for the majority of shigellosis. So one of the first things we looked at was the population. We wanted to look at the population structure of GEMS isolates. And to do this, we constructed maximum likelihood trees based on core genome SNPs. So the trees were generated using IQ trees. And to contextualize the GEMS isolate within the established global lineage, we incorporated publicly available data. So shown here is the midpoint rooted Sony tree. The red dots you see here are the GEMS isolates, and the blue dots are the publicly available data. And we can see the tree revealed that Sony has a really limited genetic diversity, in which we saw all but 2 of the GEMS isolates belong to this lineage 3 here. And this lineage has been previously established as the globally dominant Sony lineage. So this lineage is known to be to cause greater burden of disease than compared to any other Sony lineages. And then lineage 3 further divides into further sublineages. But for the purpose of this talk, I'm just going to call them lineage 1 and 2. So across both sublineage 1 and 2, we observed geographical clustering of the isolates from each gem site, with some phylogeographic admixture from India and Bangladesh, so suggesting possible transmission between the 2 countries. And then we assessed the genomic diversity of flexneri isolates. So in contrast to Sony, we saw that Flex was more genetically diverse. So shown here is the unrooted Flex tree. And we saw that these isolates belong to 6 distinct phylogenetic groups. And then we overlaid the geographical location, and then we saw that each gem site was populated by isolates from several distinct phylo groups. So the pie chart, the area of the pie chart you see is proportional to the number of isolates contributed from each site. So you can see most of the isolates came from Bangladesh. The ratio of the pie chart shows the composition of the isolates from different phylo groups. So in most region, we can see that isolates from phylo group 3, which is this like yellow phylo group here, they're the most prevalent, and they cause the highest disease burden across most gem sites. And then we wanted to assess the prevalence of resistance to help inform a treatment with antimicrobial therapy. And to do this, we screened for presence of genetic determinants that confer antimicrobial resistance. So the bar charts that you see here, they show percentages of Flex and Sony isolates from gens that contain various AMR genes. And these were grouped based on different drug classes. The isolates were also grouped into different geographical origins. So the green bars represent isolates from Asia, and then the yellow bars are from isolates from East Africa, and then the blue bars represent isolates from West Africa. And our analysis showed that more than 60% of the GEMS isolates are multi-drug resistant. So they carry genetic determinants that confer resistance to more than 3 classes of drugs. So for both species, we detected a mutation that confers reduced susceptibility to ciprofloxacin. And this is the current recommended antimicrobial therapy for shigellosis. And these mutations were geographically were identified in isolates that were geographically restricted to Asia. These are de novo mutations at the quinolone resistant determining region. And when we look at the phylogenetic distribution of these isolates, it shows that these mutations were independently acquired across multiple phylo groups, which shows that there's selection due to heavy usage of these drugs in these regions. So with the worrying increase in resistance, vaccination is a good approach for management of shigellosis. So currently there's no licensed vaccine available. but there are several in development, and we wanted to utilize this data set to assess the diversity of a few of the vaccine candidates and how that may impact its likely effectiveness. So for protein antigen candidates, we assessed how conserved they were within the GEMS dataset, and this was done through homologous sequence search. So based on 80% amino acid sequence identity. And for FLEX, with the exception of SIG-A, we saw that more than 80% of the isolate carried these vaccine candidate, which was good. But for SONI, either B, C, and D, and this ICSP protein, they were absent in majority of the isolates. But we think this is mostly due to the fact that these genes are carried on the virulence plasmid, and this plasmid in SONI is known to be really unstable during laboratory conditions. So it's often lost during subculturing. So I think this is why we see the low carriage. We then assess the amino acid sequence diversity of these antigen candidates. So you can see for IPA, B, C, and D, we observed about 4 to 8% amino acid sequence variations. And some of these variations were identified in known epitopes, which is like these colored blocks here. So this is a problem because such variation might impact the effectiveness of these antigen vaccines, and we should carefully evaluate them. We also saw low levels of amino acid sequence variation in the other 3 candidates. Okay, so protection against Shigella it appears to be mediated by immune response that's serotype specific to the Shigella O antigens. So the different O antigen structures give rise to different serotypes, and shown here are just some of the different O antigen structures and the serotype that it determines. So for SONI, there's only one structure, so there's only one serotype. But for FLEX, we know that currently there are 15 serotypes. And serotyping analysis from the GEMS dataset shows that multiple serotypes contribute to Shigellosis at the different sites. And if we were to develop a multivalent vaccine, they should include some of the most predominant serotypes. But one really important thing to note here is that the modification of these O antigens and FLEX that determines what serotypes they are, they're carried on genes. They're performed by genes that's carried by bacteriophages, and sometimes they're also carried on plasmids as well, on these serotype determining regions. So these elements are mobile, they allow modification of O antigens, and in cause it leads to a serotype switch. And this is a major strategy that's used by FLEX to evade host immunity. So we wanted to determine how prevalent serotype switching is from the GEMS dataset. Okay, so here is the FLEX phylogeny, and then on the outer here, the color strip, they represent the different serotypes. And we do see a pretty good correlation of the phylogenetic groups with serotypes, but we can also see that, you know, where the change of colors are, they represent a serotype switch. It's not, it doesn't appear to be very prevalent. And we didn't really see any association of serotype switching with a particular phylogroup. But nevertheless, we still want to, in the future, perform a temporal analysis to try and estimate that time frame of serotype switching, so we can help to inform effective vaccination programs to basically estimate this anticipated time frame of like when we can expect a vaccine escape due to these serotype switching events. Okay, so GEMS was a case control study. So children were enrolled with moderate to severe diarrheal disease if they had diarrhea, and GEMS defined diarrhea as having more than 3 loose stools for over 24 hours. They also included different clinical presentations, such as sunken eyes and a few other definitions. And also if these children required treatments or hospitalizations at the health centers. And the controls were 1 to 3 matched to the cases. And they comprised of randomly selected age-matched community children without diarrhea. So we did some genome-wide association studies, and these were on the FLEX dataset. So we have a total of 513 cases and 72 controls. And we wanted to perform GWAS just to look for genetic signatures that were associated with disease, with case isolates, so isolates that have disease outcome. So the analysis was done using Picea, looking for enrichment of SNPs that were associated with case isolates. And shown here is the Manhattan plot displaying all the Picea hits. The most significant hit that we identified was a SNP that was located in an intergenic region. But we also identified, we saw lots of hits on this large virulent plasmid. So this is a virulent plasmid that's around 200 kb and it encodes for a lot of essential proteins that's essential for invasion, intracellular survival, and propagation of Shigella. We also looked for presence and absence of genes that were associated with disease. And for this analysis, we ran both Picea and Scory. Picea identified more significant hits than Scory, but there were 25 overlapping genes that was identified from both analysis. And within these 25, 13 were hypothetical, and then the 13 annotated, and the rest of the annotated genes are shown on this table here. So shown here again is the flex tree, and we overlaid the case control metadata on the outer ring here. So the lighter pink that you see are isolates derived from control children, and the darker pink are isolates derived from case children. And control in this case represent asymptomatic Shigella isolates. So we noticed that isolates from phylogroup 1 and 2 here, they seem to have a higher percentage of control isolates than compared to the other phylogroup, and this was significant. So phylogroup 1 and 2 was found to be significantly associated with having control isolates. And then we overlaid the presence of the genes that was identified by both Picea and Scory. So these are the genes that were annotated. And it was apparent that these genes are enriched in case isolates that's specific to phylogroup 1 here. And we found that all but 2 of the 25 enriched genes, they were carried on the virulence plasmid. So shown green in the outer ring here is the presence of the virulent plasmid, which corresponds to pretty well with the carriage of these genes identified. So then we ran a chi-square test to look for association of the presence of the case of the virulent plasmid to case isolate, and it was significant. And yeah, so we did see that more than half of the isolates from this phylogroup was missing this virulence plasmid. And this is really interesting for us because this virulent plasmid is, or traditionally is almost considered part of the core genome, which is really essential for Shigella to cause disease. And usually, you know, in studies, when we see that there's a loss of plasmid, we just assume that it's a laboratory artifact and that it was lost during subculturing. But we've shown here that, you know, it is possible for isolates to circulate in this population without the plasmid. And not having this plasmid may be a genetic factor that was associated with possible asymptomatic isolates, like that's specific from phylogroup 1. So yeah, results from this study, we've shown that demonstrated the important usage of whole genome sequencing analysis of a representative data set to provide insights to Shigella causing the highest disease burden of disease. And we revealed that at each gem site it was comprised of genetically diverse population of flexneri, which may complicate both conserved protein antigen and also serotype specific vaccination approaches. And we also observed a worrying level of flexneri and Sony isolates that were carrying genetic determinants, conferring reduced susceptibility to ciprofloxacin, which is the current recommended antimicrobial treatment for shigellosis. And this finding highlights that we really need to find an alternative way to manage this disease. And also for GWAS, we identified presence of genes that are carried on the virulence plasmid, which might be associated with asymptomatic controls isolates in phylogroup 1. Yeah, and thank you so much for everyone for tuning in. That's it.
All right, thank you so much, Rebecca. That's great. We have a lot of questions, and we do have a couple of minutes for them, if you feel up for it.
Yeah, sure.
Let's see. Okay, here's an easy one from Evan. Did you identify plasmid replicons and their contribution to the AMR in this population? I guess you did that.
Yeah.
Yeah.
Yeah. Sorry, can you say that plasmid
So the question was
replicons?
about plasmid replicons and any link with AMR in
Yeah. No, actually, we haven't really looked at that yet. But yeah, that's a good suggestion. Maybe I can dig into that deeper. It's so with Shigella, it's a bit tricky because it's just the genome is full of insertion sequences, so it's really hard to construct a whole plasmid or genome. So we get contig fragments, but we can never say that it is coming from that particular plasmid. So it's hard to know if we see an AMR and it's associated with that plasmid replicon.
Mm-hmm. Yeah. Another question was, so do you think Shigella is evading host immunity due to GTR-ABS-based O-antigen modification, or is it evasion of phage predation and the presumed host immunity evasion as a side effect, is just a side effect? That is very tough. So what do you, yeah.
Yeah, I'm gonna have to think about. I'm gonna have to think about that one and get back to you. I'm not sure.
Well, I hope you're on the Discord to take on some of these more meaty questions.
Yeah, definitely.
I think we've got time maybe for one more. How would a potential vaccine for Shigella affect carriage rates of commensal E. coli given their genome similarities? And I also had a question along those lines, which is, given what you presented, how much of it is applicable to enteroinvasive E. coli?
Yeah. I mean, yeah, I haven't looked too much about. Not sure about that one.
That's all right. Okay, well that's good. But
I'll have a think. I'll have a think about those questions and then get back to you.
Yeah. Yeah, of course. So that's what the Discord's for. So yeah, if anyone has any extra questions, we can move it over there and have some discussion over some virtual coffee, virtual beer. And so yeah, I'd like to, again, on behalf of everyone tuning in, there's a good portion of people listening in right now and over the session. I want to thank all of the speakers today for some really wonderful talks. And yeah, thanks everyone for attending. We're going to switch over to the